Great, we're here at Big River Chestnuts in Sunderland, Mass. This is a 10-acre farm on the floodplain of the Connecticut River. We're right next to the river on some deep silty soils, and we're growing chestnuts. So you can see some of the chestnuts in the tubes here that are now 4 years old. They're really just getting established and just coming into production. So we're pretty excited to begin producing chestnuts here. We're also doing small fruit in the alleys. So we've got elderberry, Aronia, black currants, primarily in the alleys between the chestnuts. We've also got chickens, meat birds that we're arranging in the alleys and moving around the field. And all of that is part of the process of improving the soils. Part of our plan, our soil health practices that we're using. One part of the project is that we're focused on how does this agroforestry, integrated agroforestry system improve the soils over time, and seeing how the root interactions, the no-till, in addition to a bunch of different practices that we're using, how does that help improve the soil over time, and how does it build the soil biology that is really doing a lot of the work of the soil health that we're seeing. And so some of the things we're doing, of course, it's a no-till system with the perennial crops. We're using a lot of mulch, such as these wood chips here. We're adding this kind of biomass that is going to feed the mycorrhizae, the fungi. We're inoculating with other kinds of soil biology with some different kinds of amendments. And we're adding mineral amendments to try and balance and build the mineral levels that have been depleted over time. We're doing subsoiling, physically reducing some of the compaction that's there, and then we're adding cover crops that will come in and do some of that digging into the compacted layers in the soil, breaking up the density and adding roots down deeper. So those are some of the things that we're doing, and then we're tracking the changes over time with soil testing. So we've got some different fertility tests as well as biology, as well as just some of the physical parameters like the density, carbon levels, and trying to understand how those changes happen over time in this kind of perennial cropping system.
Hi there, I'm Monique Bosch. I'm Soil Health Lead for CT NOFA, and I'm so happy to be here at Big River Chestnuts Farm in Sunderland, Massachusetts, working with Jono and Stephanie today. We are going to be getting samples from various parts of the fields here. We're going to be looking at the soil around the chestnuts and also around the Aronia, some of the cover crop, and we're looking for a number of things. We want to see what the soil structure looks like. We also want to see what the life looks like. So looking at it under the microscope, you really, definitely get a sense of the microbial activity in terms of bacteria, the relationship or the ratio between bacteria and fungi. Are there any predators, larger predators that complete the soil food web?
So this is Stephanie Hartzig, who's helping out with our assessment of the soils and examining the data and working with us as an intern for the fall.
So for this internship, I'm here to get an idea of what this type of system looks like whenever you have trees incorporated with perennial crops, to understand how agroforestry in general can be a better way of doing agriculture than monoculture or other traditional means.
All right, here we go. This is our block of Aronias. We're producing these as a small fruit, a nutritive, super high nutrient small fruit, and we'll be selling the whole fruit frozen, and had our first crop this year in year 2. And we're expanding slowly and establishing these using this kind of mulching with wood chips that we get from a arborist down the road. By adding these here, we're bringing in the substrate that the fungi can colonize and begin to utilize, and they're going to be the doing a lot of the work in the soil for improving the conditions. And we can already see some changes in the top layers of the soil where we're getting some nice partially decomposed organic matter. And it's reducing the weed competition somewhat, but we don't really mind too much. There's some dandelion here, there's some clovers, and those are all actually beneficial because they're rooting into the soil. So we don't necessarily want to get rid of all the other plants. We want to reduce some of the competition, and by bringing in the mulch, we're just feeding the system completely. So we're going to be testing for the soil biology here under the Aronia, and Monique Bosch from Connecticut NOFA is here taking some samples that we'll look at under the microscope.
So here we are, sample 1. We're looking at soil around the Aronia, and we found this beautiful piece of fungal hyphae. We're also seeing flagellate activity. Here's a nice looking aggregate. It looks quite sandy. Those tiny glass bits are some sand. If we zoom into 400 times, so you can see all of that bacterial activity. So you've got aggregates being formed and this really large fungal hyphae. Nice coloring, mature, and those are the aggregates, those copper colored pieces.
We're now in the south side of the field, the part of the field where we've been rotating chickens through, fairly large numbers, blocks of 300, doing a couple thousand a year for the last several years. So a lot of manure added in a rotational grazing kind of program. And it's led to some very lush growth conditions even in some of these cover crops like the daikon here that we've sowed in, and some of the wilder plants like the Canada thistle. So what we're going to do is pull up some of this daikon, and we're going to take a look at the soil biology activity around those roots. Okay, so we're taking a look at the soil. We're seeing what kind of root activity. Here's the clover with some really nice nodulation, some nitrogen fixing nodules which are in association with bacteria. Daikon radish, so that's pushing into the soil and breaking up that compacted layer. This is a little one. They'll get quite large and are also tasty as well.
So let's get a soil sample from the daikon radish here. "40 times magnification, so very consistent. This is the soil under the daikon radish on the north side. So we're seeing there's a variety of bacteria here. There are rods and round cocci. We're seeing sand and silt. We'd like to see more fungal in here, but there are aggregates being formed. I go up to 400, you can see those nice aggregates there. That small, tiny piece of fungal hyphae there. But it kind of makes sense because this is where the cover crop is, and from the history, this had been growing annual vegetables for generations, so it would explain why you don't necessarily have the fungal in here. But I do like seeing this aggregate structure and the very active bacteria.
Here in this cover crop patch, there's been a lot of chicken activity, got some soil development in the topsoil, some of the biomass, and also a little mushroom. It's looking pretty good. You know, it's starting to develop some topsoil and the conditions are starting to change. So, all right, so we're gonna dig down here, and we're at the base of this chestnut tree. We're going to go down about a foot down and get a sample to see what kind of activity is happening down in the upper root zone of this tree.
Okay, so I'm getting a sample just around those active roots there. 40 times magnification. This is under a chestnut tree, 4 inches down on the north side. And we found this nice soil structure here and a lot of fungi and aggregates in this one particular piece. This is now 400 times magnification. So I like the soil structure here, those aggregates, these nice dark brown fungi. And we're also seeing some more clear pieces of fungi in here. 100 times magnification. This is the chestnuts 1 foot down on the north side. So we're seeing really good aggregate structure. We found this one fungal hyphae that just goes on and on. So that's good to see, especially looking at such a wide angle. I thought I saw another piece of fungal hyphae there. Maybe this one here. Totally different kind, so you have diversity of fungi. We've been seeing flagellates in here, but I would like to see more fungi. Although these could be bits of fungi in the top. So whenever you see those smaller pieces. But I like the soil structure. I like all these aggregates. You can see the sand particles, the clear pieces.
All right, so we're gonna dig another test hole under this tree, just in a different side of the field. I can tell there's some good rooting here because I can feel some of these roots. Pretty good sized root coming off of this tree.
40 times magnification. This is the chestnut on the south side, 4 inches down. We're looking at 40 times magnification. And I'm just going to zoom in here 100 times magnification. Really wonderful aggregate structure in this. There's a lot of bacteria. I'm not seeing the fungi that I would like to. I'll zoom into 400 times magnification. You can see how dense it is with the aggregates that are made up of bacteria. 40 times magnification. This is a chestnut on the south side. We've gone down a foot now, where we found some roots right along the drip line. And again, really good development of aggregates down here. The theme of not finding the fungal that I would like to see. But let's just zoom in on this. Very consistent throughout. This is 100 times magnification. I think I'm seeing quite a bit of movement, which makes me think that there are flagellates in here. Okay, so if I go to 400 times magnification, you can see it's teeming with bacteria. You have some aggregates going on. It could be a small piece of fungal hyphae in the center there. So these are new trees. They've been in 3 or 4 years. So hopefully those small fungal strands will start becoming more mature. But nice aggregates and plenty of bacteria throughout.
So that was really great, looking at the microscopy and the activity in the soil, and it really is exciting to see, and it looks like there's a lot of life in the soil. So we're starting to really get some good activity, good soil structure, and the aggregates are coming together.
So for me, this was really interesting to see all the life under the microscope, see it happening in real time. And for my internship, it really confirms the fact that the agroforestry is a really great option for creating soil biology health. It with the integrated agroforestry system that we have here, which means trees and shrubs and different crops planted together, as well as a little bit of silvopasture with the grazing chickens, that we're finding the more diversity you have above the soil, you'll have more diversity below the soil.
We're looking at ways to increase the fungi activity in the soil, and some of the things we've been doing, such as adding wood chips and chopping back the biomass to get that material on the ground, are good options for feeding the fungi. And it's just going to take some time. We're seeing some beginning changes. We're seeing some fungi in the wood chips at the surface, and the microscope work is just beginning to show us that the changes are happening, but it needs a little bit more time, and we're in it for the long haul with this kind of system, and so excited to see the changes starting to happen. Seeing if we could tell differences in the soil depending on some of these different practices that are being used, and at this point we are doing monitoring of the growth of the trees, the condition of the trees, which ones are starting to come into bear sooner, setting more nuts on them. But we haven't been able to tell any really big differences in the tree growth. We are seeing differences in the cover crop growth and the understory and the lushness of the south field that has all that manure being laid down at the surface, but not any big differences in the tree growth at this point. So that really is our goal, to sort of balance out the wet, dry changes, keep that soil covered, keep the cover crops going, and even this kind of perennial pasture mix that we're moving towards under the trees and the shrub crops. We would want to have as much life happening throughout the season, even into the winter, and then continuing to add wood chips and some spot treatments in areas where we feel like there's some exposed soil, or we want to increase the carbon more quickly.
In conclusion, I had an amazing internship here. John, it was such a pleasure to work with. I couldn't have, chosen a better spot to spend 3 months in fall. I've learned so much, and I'm really excited to take what I've learned here about soil health, soil biology, agroforestry, and go to the next place and really understand further and take what I've learned to share it and hopefully inspire others to take the jump and start creating more of these systems.
I think we've learned a lot. We're really establishing a lot of understanding of the baseline conditions here at the farm in these soils. We're learning a lot about how the different practices can improve the soil, some faster, some slower, and being able to track those changes over time, I think, is going to be super helpful for other farmers and other growers as they get set up and want to overcome challenges, soil health challenges, and look to build better soil health in the future, because that's really what it's all about.